In the South Pacific, sometime past midnight, a net breaks the surface after being pulled up from deep water. It has been down for hours, long enough to reach depths where light does not exist. What comes up is not something the crew can immediately place. That is not as unusual as it should be. Squids have been on this planet for longer than the dinosaurs. They were here before the first flower, before the first bird, before almost everything alive today. And in all that time, hundreds of millions of years, they never stop changing. What they have become in some parts of the deep ocean is difficult to describe. In 2004, a team of scientists lowered a camera into the North Pacific and left it in total darkness for hours. Baited, waiting, what eventually came to it became the first confirmed footage of a giant squid alive in its natural environment. Up until that moment, everything we had, every drawing, every measurement, every description, came from dead bodies and the contents of whale stomachs. That one piece of footage changed the question entirely. Not whether they exist, but what they are actually doing down there, in the dark, at depths where the pressure would kill a human being in an instant. And the giant squid is not even close to the strangest one. There are species alive right now with eyes the size of a dinner plate, the largest eyes of any animal on Earth. There are squids that do not release ink when threatened. They release light. There are species we have never seen alive, only identified from beaks found inside dead whales, from animals that were clearly enormous, that clearly existed, and that no one has ever directly observed. Today we go through some of the strangest squids ever brought up from the deep, and what they suggest about life far below the surface. But before we do, let me know in the comments. Are you listening to this to fall asleep, or you're here only for the knowledge? If this kind of topic is something you find interesting, consider subscribing and leaving a like. It helps the channel more than you might expect. Now let's go down. Light does not vanish all at once. It thins, fades, and withdraws in measured layers, each one reshaping the rules of existence as it recedes. Near the surface, within the first 200 meters, the ocean holds what is familiar. Sunlight penetrates freely, feeding microscopic plants that turn light into energy. This is the bright skin of the sea, where movement is visible and color still has meaning. It is the version of the ocean most often imagined, a place where life is driven directly by the sun. Below that, from 200 to 1,000 meters, light begins to fail. The world shifts into a dim blue haze where illumination lingers but no longer supports growth. Here, visibility becomes uncertain and forms dissolve into silhouettes. It is a transitional space where creatures rise and fall in vast daily movements, following cycles that are no longer tied to direct sunlight, but still shaped by its distant presence. Deeper still, from 1,000 to 4,000 meters, light disappears completely. This is not darkness as experienced on land, where the memory of light remains. This is a condition without illumination of any kind, where the concept of brightness has no relevance. In this depth, vision becomes something entirely different, adapted to signals that are no longer part of the surface world. Beyond that lies the abyssal zone, stretching from 4,000 to 6,000 meters. The water here hovers just above freezing, held under immense weight. Pressure builds steadily, with descent increasing by 1 atmosphere every 10 meters. At 1,000 meters, each small area of surface bears a force comparable to the weight of a large motorcycle. By the time the abyss is reached, that force has multiplied many times over, shaping every structure that can exist there. Below 6,000 meters, the ocean narrows into trenches that cut deep into the Earth's crust. This is the Hadal Zone, a realm defined by depth itself, where even the seafloor becomes fragmented and steep. The deepest confirmed observation of a squid has occurred at 7,000 meters within this confined and extreme environment. In all of these depths beyond the reach of light, energy no longer arrives from above in a direct form. Instead, it descends slowly. Tiny fragments of organic material drift downward from the surface, a continuous fall of particles that accumulate over time. This steady rain, composed of remnants from life far above, forms the base of an ecosystem sustained by what has already lived. Everything that exists here is shaped by absence. No photosynthesis occurs, no direct sunlight reaches, and no simple visual cues remain. Life continues, but under conditions that demand different solutions at every level, from the structure of cells to the patterns of behavior. Squids did not simply enter this environment and persist. Over hundreds of millions of years, they became part of it, altering their bodies and systems to match conditions that would break most other forms of life. Their physiology reflects pressures, temperatures, and chemical balances that are far removed from the surface world. There are 800 squid species described by science, each one a result of this long adaptation. The number that remains unseen is likely far greater, existing within layers that are still only partially understood. Long before forests spread across continents, before bones hardened into limbs that could bear weight on land, something soft and fluid was already moving through ancient seas. It left behind almost no trace of its body, only fragments of mineral shell and subtle imprints pressed into stone. Yet those traces stretch back 500 million years, reaching into the earliest chapters of complex life. Cephalopods emerge in the fossil record during the Cambrian period, around 500 million years ago. At that time, life itself was still experimenting with form. Eyes were new, movement was becoming deliberate, and the oceans held organisms unlike anything that would follow. Within this early expansion, cephalopods began as shelled creatures, their bodies anchored within rigid structures, drifting or slowly propelling themselves through water that had never known vertebrate motion. As time continued, the world above the ocean changed slowly. Then suddenly, the first animals stepped onto land around 375 million years ago, transforming coastlines into new frontiers. Later came the rise of reptiles, then the age of dinosaurs. At approximately 230 million years ago, followed closely by the first mammals. Through all of this, cephalopods were not emerging, but persisting, already shaped by hundreds of millions of years of prior evolution. The earliest forms recognizable as squid appear around 300 million years ago. By then, the lineage had already undergone profound changes, abandoning external shells and developing bodies capable of swift, controlled movement. This transition allowed for a different kind of existence, one that favored flexibility over protection, speed over rigidity. It marked the beginning of a direction that would continue without pause. Across the long history of life, there have been 5 major extinction events, each one reshaping the planet with a scale that is difficult to hold in a single thought. The Ordovician extinction, around 445 million years ago, erased the majority of species then living. The late Devonian followed, then the Permian, which removed nearly all marine life in a single interval. Later came the Triassic extinction, and finally the Cretaceous event 66 million years ago, which ended the reign of the dinosaurs. Cephalopods passed through each of these thresholds, not unchanged, but not eliminated. Their survival did not depend on strength or dominance. Instead, it rested on a capacity for transformation, a way of shifting form and behavior in response to whatever remained after each collapse. Where rigid bodies failed, their softness allowed variation. Where specialization became a limit, their adaptability created new possibilities. Within this lineage, one branch held on to its ancient structure. The nautilus still carries a coiled shell, moving through the ocean in a form that is altered very little across immense spans of time. It is often described as a living fossil, a reminder of what once was. Squids followed a different path. Their evolution did not settle. It accelerated, branching into forms that occupy nearly every marine environment. Some remain small enough to fit within the narrowest spaces, while others extend to lengths that rival the largest known invertebrates. They inhabit shallow coastal waters, open ocean expanses, and depths that press against the limits of biological endurance. What exists now is not an anomaly or an endpoint. It is the current state of a process that has continued without interruption for 500 million years, shaped by cycles of loss and renewal that have never fully stopped. If the body is followed inward rather than observed from the outside, its design begins to reveal itself in quiet contradictions. Structures arranged in ways that seem almost accidental until their purpose settles into place. At the center, the brain forms a ring. It does not sit above or behind the digestive system, as it does in most animals. Instead, it encircles the esophagus completely, so that every piece of food must pass through the opening at its core. This arrangement leaves no margin for error. Anything too large cannot be swallowed without risk, and so feeding becomes an act shaped as much by anatomy as by appetite. Circulation moves differently as well. 3 hearts share the work, each one assigned a separate role. 2 smaller hearts push blood through the gills, where oxygen is gathered, while a larger heart distributes it through the rest of the body. When the animal accelerates using its jet, the main heart pauses, a brief stillness in the middle of motion. Speed can be reached but not maintained, and so movement becomes a balance between sudden bursts and quiet drifting. The blood itself carries a different chemistry. Instead of iron, it relies on copper to bind oxygen, giving it a deep blue color when oxygenated. This system performs efficiently in cold and oxygen-poor conditions, maintaining function where other systems would begin to fail. It is a solution tuned to environments that do not offer abundance. Control is distributed rather than centralized. Out of roughly 500 million neurons, only a portion resides in the brain. The majority extend outward into the arms, where they form dense networks capable of processing sensation and guiding movement independently. An arm can respond to texture, pressure, and chemical cues without waiting for instruction, creating a body that thinks in multiple places at once. Within the mantle, a single internal structure provides support. The gladius, a thin and flexible remnant of an ancestral shell, lies embedded like a translucent spine. It offers shape without rigidity, allowing the body to remain both stable and compressible, a framework that bends rather than resists. Movement emerges from the controlled exchange of water. Fluid is drawn into the mantle cavity and expelled through a muscular siphon, which can shift direction with precision. This creates thrust that can be aimed, not just forward, but in multiple orientations, allowing sudden changes in position. Fins extend from the mantle, moving more slowly, maintaining balance, and enabling gentle adjustments when speed is not required. Along the arms, contact is managed through structures that vary across species. Suckers form rings of hardened material, sometimes smooth, sometimes edged, capable of holding or cutting depending on their form. In certain lineages, these are replaced entirely by rotating hooks, mechanical and deliberate in their grip. Defense introduces another layer of complexity. Ink is stored near the digestive organs and expelled through the same siphon used for movement. Mixed with mucus, it does not disperse immediately, but holds its shape for a moment, forming a drifting outline that resembles the animal itself. In that brief interval, attention is redirected and the body moves away. The outer surface operates as a dynamic interface with the environment. Layers of cells interact to produce color, reflect light, and scatter brightness. Pigment sacs expand and contract under direct neural control, while deeper structures manipulate how light is reflected and diffused. The result is a surface capable of rapid transformation, shifting appearance in ways that match surroundings or convey signals. All of this exists as a starting point, a configuration already far removed from familiar biological patterns, yet still only an initial form shaped further by conditions beyond ordinary experience. In a layer of the ocean where breath itself becomes scarce, a small body hangs suspended, not in motion, not at rest, but in a kind of quiet balance that avoids both. The water here holds almost no oxygen, a thin chemical presence that fades to levels most animals cannot endure. Yet within this narrow band, life persists in a form that has chosen stillness over pursuit. Vamproteuthis infernalis belongs to a lineage that stands apart, neither aligned fully with squid nor with octopus, but occupying its own branch, singular and without close relatives among living species. Its existence is tied entirely to this oxygen minimum zone, typically found between 600 and 900 meters, where dissolved oxygen drops to less than 3% of what is found near the surface. In this range, muscle and metabolism falter in most organisms. Here they are reshaped. Rather than resisting the limitation, this animal accommodates it. Its metabolic rate is the lowest recorded among cephalopods, a slow internal rhythm that minimizes demand. Movement is reduced to small adjustments, gentle repositioning, rather than active swimming. It does not pursue prey, does not expend energy in bursts or chases. Instead, it waits within the drifting medium, allowing food to come to it. Its feeding method reflects this restraint. Two long filaments extend outward, almost invisible, coated in mucus that captures fine particles suspended in the water. These particles include organic debris and microscopic remnants, fragments that have descended from higher layers and settled into this region. The filaments are drawn inward periodically, delivering collected material to the mouth in a continuous, unhurried cycle. Defense follows a similar logic of economy. Ink, common among related species, is absent. In its place, the animal produces a luminous secretion, released from the tips of its arms. The substance glows with a steady blue light, persisting for minutes rather than dispersing instantly. It forms a suspended field that lingers as the body withdraws, leaving behind a point of attention that remains fixed while the animal itself moves away. A second response alters its shape. The arms, connected by a thin web, fold upward and over the body, turning inside out to reveal rows of soft spines. The transformation is not rapid, but deliberate, expanding the apparent size and shifting the outline into something less recognizable. It is a gesture that relies on form rather than force. Vision is adapted to this dim environment in a way that reflects its scale. The eyes are proportionally the largest known relative to body size among animals, capable of detecting faint signals within minimal light. Their color appears to change depending on how light interacts with them, shifting between deep blue and red tones without altering their function. The outer surface of the body carries a muted palette, capable of subtle variation between dark and pale shades. These changes occur smoothly, adjusting to the limited illumination of the surroundings, rather than creating sharp contrasts. The temperature of this environment remains between 2 and 6 degrees Celsius, stable and cold, supporting few forms of life beyond specialized organisms and microbial communities. Within this narrow range, the vampire squid continues its existence, shaped by conditions that exclude most others, maintaining a presence that requires almost nothing beyond time and the slow arrival of what drifts down. Weight gathers in this animal in a way that feels deliberate, as if density itself has been selected for over time. Its form is not extended or delicate, but compact, each part built with a thickness that resists easy comparison to other squids. It carries mass as its defining quality, a presence shaped less by length than by substance. Mesonicothothus hamiltoni stands apart for this reason. It is not the longest of its kind, yet it outweighs all others by a wide margin. The largest confirmed specimen, brought up in 2007 from the Ross Sea near Antarctica, reached a mass of 495 kilograms with a mantle measuring 2.5 meters. Its proportions give it a different silhouette, broader and heavier, where others appear drawn out and narrow. This distinction becomes clearer when placed beside its distant relative, the giant squid. That species can approach a similar mantle length, around 2.25 meters, yet its body remains comparatively light. The colossal squid compresses that scale into a denser structure, emphasizing strength over reach, volume over extension. Its eyes mark another boundary. The specimen retrieved in that same year possessed eyes measuring 27 centimeters across, wider than a human head. These are the largest eyes ever recorded in any living animal. Their scale is not ornamental but functional, an adaptation to a world where detection depends on the faintest disturbances. The leading interpretation suggests that these eyes are tuned not to constant light, but to brief signals created by movement. A large body passing through dark water can trigger subtle flashes from surrounding organisms, a cascade of small reactions that trace its path. A predator moving below leaves behind a shifting pattern of illumination, one that can be perceived at distance if the eye is capable of gathering enough of that scattered light. In this way, the presence of another animal becomes visible not directly, but through the response it produces in the environment around it. The arms and tentacles extend this emphasis on force. Instead of smooth suction structures, the tentacle clubs carry rotating hooks, each one capable of turning in place. These hooks are embedded within the tissue, forming a gripping surface that can secure contact in a different manner than suction alone. Marks consistent with these hooks have been found on the skin of sperm whales, leaving patterns that differ from the circular impressions associated with other species. The environment in which this animal lives is defined by cold. Antarctic waters maintain low temperatures that influence every aspect of its biology, from muscle function to metabolic rate. The Ross Sea serves as a central region where encounters have occurred, though direct observation remains limited. Nearly all information comes from individuals brought up unintentionally during fishing operations, captured at depths where long lines are set for other species. No adult has been filmed alive within its natural setting. The absence of direct observation leaves its behavior inferred from anatomy and indirect evidence. Fewer than 10 complete adult specimens have been examined in detail, each one contributing a fragment to an incomplete picture. What can be understood emerges through these fragments. A body built for power rather than speed, vision adapted to signals that travel through darkness, and tools for grasping that operate through mechanical engagement. It exists within a narrow range of conditions, shaped by cold and depth, carrying a form that reflects both constraint and possibility without ever fully revealing how it moves through its own world. For a long time, This animal existed as an outline assembled from fragments, a form inferred rather than witnessed. It appeared in pieces, in what remained after encounters had already ended, leaving behind only evidence that something large had passed through unseen water. Architeuthis ducks entered scientific record in 1857, when Danish naturalist Japetus Steenstrup described it from a beak recovered from a stranded body. The description gave it a name and a place within classification, yet it did not provide a living image. What followed was more than a century in which the animal remained present in knowledge but absent in observation. Specimens surfaced occasionally, carried ashore or caught in fishing equipment, often damaged, often incomplete. Others were discovered within the stomachs of sperm whales, reduced to hard structures that resisted digestion. From these remnants, size could be estimated, proportions suggested, but behavior remained entirely unknown. The body could be measured, but not understood in motion. During this period, the creature expanded beyond science into narrative. Maritime cultures across northern and western Europe had long carried accounts of immense beings beneath the sea, forms that rose from depth and altered the surface itself. In Scandinavian, Icelandic, and Basque traditions, the kraken took shape as something vast and elusive. It appears in the 13th century Icelandic text Urvar Odd's saga, described not with precision but with scale, an entity capable of creating currents and disturbances that reshaped the water around it. These accounts did not align with measured specimens, yet they persisted, overlapping with scientific knowledge in a way that blurred boundaries between observation and imagination. For 147 years, the giant squid occupied this space, partially documented, partially mythologized, never fully resolved. That changed on September 30th, 2004. In the North Pacific, near the Ogasawara Islands, a camera was lowered to 900 meters, attached to a long line with bait. It remained there for hours, recording in a depth where light no longer defined form. Over the course of that time, it captured 556 images of a living adult giant squid. The sequence revealed an animal in motion, approaching and interacting with the bait repeatedly. It did not drift passively. It engaged with force, extending its tentacles, striking and pulling. During the encounter, one of its tentacles was severed and later recovered, confirming the identity beyond uncertainty. Several years later, in 2012, video footage was obtained at a slightly shallower depth, around 630 meters. For the first time, continuous movement could be observed. The animal appeared active and responsive, advancing through the water with directed intent. It did not resemble the slow, weakened forms previously studied on land. It moved with precision, each motion controlled, each interaction purposeful. Its arms and tentacles carry rings lined with small, hardened teeth, structures capable of leaving distinct marks. Scars observed on sperm whales correspond to these rings, some measuring up to 5 centimeters across. Other scars exceed this size, suggesting encounters with individuals larger than those directly recorded. Females reach the greatest dimensions, with confirmed lengths between 12 and 13 meters. Reports of larger sizes exist, derived from indirect evidence rather than complete specimens, extending the range beyond what has been physically measured. The image that emerges is no longer assembled from fragments alone. It includes motion, interaction, and presence, forming a more complete understanding that replaces absence with observation, while still leaving much unseen within the depths it inhabits. There are places in the ocean where concealment cannot rely on structure, where nothing stands between a body and the surrounding water. In these spaces, visibility becomes a problem without edges, a condition that cannot be solved by retreat or cover. Within this open water, a lineage of squid has followed a different path. The glass squids, belonging to the family Cranchidae, have altered their bodies toward near absence, reducing their visibility not by blending into surroundings, but by removing the visual cues that define a solid form. Their tissues hold very little pigment. Muscles, mantle, and much of the internal anatomy allow light to pass through with minimal distortion, leaving only faint outlines that shift with movement. Transparency, however, is not absolute. Certain organs resist this transformation. The digestive gland, liver, and eyes depend on molecules that interact with light in ways that cannot be made clear. These structures remain opaque, creating interruptions within an otherwise transparent body. In the dim environment where these animals live, even a small obstruction can cast a detectable shadow. The problem becomes directional. In the midwater zone, illumination arrives from above as a diffuse, filtered glow. Predators positioned below scan upward, detecting silhouettes against that faint brightness. Any object that interrupts the passage of light creates contrast, and contrast becomes visibility. Glass squids address this by altering orientation. Their eyes, instead of facing forward, rotate downward so that the pupil no longer blocks the light coming from above. This repositioning reduces the shadow cast upward, allowing the body to remain less distinct when viewed from below. The solution is precise, adjusting not the presence of the eye but its alignment relative to the dominant light source. This configuration appears across multiple species within the group, arising independently in separate evolutionary lines. The repetition of this trait suggests a consistent pressure shaping form in similar ways across different branches. Where light behaves predictably, adaptation follows with equal precision. Additional modifications refine this effect. Some species develop reflective layers over their opaque organs, structures that mirror the faint illumination from above. These layers scatter incoming light in a way that reduces contrast, making the organs less distinguishable even at close range. The body becomes not fully invisible, but increasingly difficult to isolate from the surrounding water. Buoyancy is managed without constant movement. In certain species, including Lecia pacifica, the internal cavity contains a solution rich in ammonium chloride. This fluid has a lower density than seawater, allowing the animal to maintain position without active swimming. The body remains suspended, neither rising nor sinking, conserving energy in an environment where resources are limited. The same solution carries an additional effect. Its chemical composition makes the tissue unpalatable to potential predators. This deterrent requires no action, no expenditure, existing as a passive quality embedded within the body itself. Development begins closer to the surface, where larvae drift in upper layers before gradually descending as they grow. Throughout this progression, the characteristic transparency remains, preserved across stages of life. The form does not change toward visibility, but maintains its reduced presence even as size increases. In this way, the glass squid does not hide in the conventional sense. It does not seek shelter or create distraction. It alters the conditions by which it is seen, adjusting structure, orientation, and internal composition so that detection becomes less certain, less immediate, and sometimes absent altogether. A body can divide its attention without dividing itself. In this species, perception is split along a single axis, with each side tuned to a different condition, as though two separate sensory worlds have been joined within one form. Histiotuthis heteropsis carries this division in its eyes. One is large, the other significantly smaller, an asymmetry that appears at first glance like imbalance. For many years, it was treated as an irregularity, a repeated flaw preserved across individuals. The consistency of the structure suggested something else, yet its purpose remained unclear until observation in natural conditions revealed how the body moves. When seen in motion, the animal does not orient itself horizontally. It maintains a steady tilt, approximately 45 degrees, with one side angled upward and the other downward. This position is not occasional. It is sustained, shaping how each eye interacts with its surroundings. The larger eye is directed toward the faint light descending from above. It contains a yellow lens that alters the incoming wavelengths, filtering out much of the blue scatter present in midwater. This adjustment increases contrast, allowing darker shapes to stand out against the diffuse glow. Objects passing between the eye and the distant surface create subtle interruptions in the light field, and the lens enhances their definition. The smaller eye faces downward into a region where ambient light no longer defines space. It lacks the filtering layer found in the larger eye, instead remaining broadly sensitive to any available illumination. Its function centers on detecting brief emissions, isolated points of light that appear and vanish within the surrounding darkness. These signals originate from other organisms, each one a momentary event that can be registered if sensitivity is sufficient. Together, the two eyes form distinct instruments. They are not variations of a single design, but separate solutions, each aligned with a specific direction and a specific type of visual input. One interprets silhouettes against a faint background. The other registers discrete flashes without reference to ambient light. The brain receives both streams simultaneously, integrating information from above and below without requiring the body to reorient. This arrangement extends beyond simple detection. The yellow lens of the larger eye reduces interference from scattered wavelengths, refining the clarity of outlines that might otherwise remain indistinct. The effect resembles an optical adjustment that isolates movement from background noise, allowing the passage of another organism to become more defined. Despite this specialized system, the species remains part of a larger food web. It is frequently identified within the stomach contents of sperm whales, indicating that its adaptations do not remove it from predation. Instead, they place it within a set of interactions where detection and avoidance operate alongside vulnerability. The body maintains its angled position as it moves, preserving the orientation of both eyes relative to their respective fields. This consistency suggests that the posture itself is integral to the visual system, not an incidental behavior but a necessary alignment. Within a single organism, two different visual conditions are processed at once, each eye gathering information from a separate direction, each adapted to a different form of light. The result is not a compromise, but a pairing, a way of perceiving that does not merge the two environments into one, but allows both to exist together without overlap. Along certain coastlines, there are nights when the water does not remain dark. It gathers a quiet glow, spreading across the surface in a way that does not reflect the sky, but seems to come from within the sea itself. This light originates from Wattisenia scintillans, a small squid measuring only a few centimeters in length, with a mass lighter than many fruits found on land. Despite its size, it produces a display that extends far beyond the scale of its body, transforming entire stretches of water into fields of shifting blue. Each year, between March and June, vast numbers of these squid move upward from depths of around 200 meters toward the shallower waters of Toyama Bay in Japan. This migration is tied to reproduction, drawing individuals into a concentrated area where spawning will occur. As they gather, the cumulative effect of their light becomes visible from the shore, a phenomenon observed for centuries before its origin was understood. The illumination is generated through specialized organs called photophores distributed across the body, including the arms, mantle, and regions near the eyes. Within these structures, a chemical interaction takes place between a molecule known as luciferin and an enzyme called luciferase. When these components combine, energy is released in the form of visible light. The process operates independently of external illumination, allowing the squid to produce light even in complete darkness. This mechanism parallels that found in fireflies on land, though the two evolved separately. The similarity arises not from shared ancestry, but from the constraints of chemistry, where certain reactions provide efficient ways to generate light within living tissue. In both environments, distant in space and separated by evolutionary paths, similar solutions have emerged. The visual system of this species reflects its use of light. Unlike most cephalopods, which rely on a single type of photoreceptor, the firefly squid possesses three. This arrangement allows it to distinguish between different wavelengths, perceiving variations in color that are otherwise inaccessible to many organisms in similar environments. The light it produces is not only emitted, but also interpreted, forming part of a communication system that operates within a narrow spectral range. Bioluminescence serves several roles within this system. Along the underside of the body, light is produced in a pattern that matches the brightness of the water above, reducing the contrast that would otherwise reveal its outline when viewed from below. This technique, known as counterillumination, allows the squid to blend into the faint light descending from the surface. At the same time, flashes and patterns are used in interactions between individuals, contributing to mating behavior. Light also functions in attracting smaller organisms, drawing them closer within reach. After spawning, the life cycle concludes. The females release their eggs and then descend, their bodies becoming part of the environment they leave behind. As they settle, they contribute organic material to the deeper layers beneath the bay, transferring nutrients downward in a process that continues across generations. Toyama Bay has been recognized for this recurring event, designated as a special natural monument in 1956. The designation acknowledges not only the visual phenomenon, but the continuity of a process that has persisted across long spans of time. A small organism, repeating a pattern each year, alters the appearance of an entire coastal region, creating light within water that would otherwise remain unlit, sustained by a cycle that continues without interruption. There are moments in recorded footage where recognition arrives slowly, not as certainty but as hesitation, a pause before understanding settles into something stable. In those frames, a shape appears that does not align with expectation, holding its form in a way that seems almost incomplete, as though part of it has been arranged according to a different set of rules. The genus Magnapinna first entered scientific description in 1998 based on juvenile specimens that resisted placement within known groups. Their proportions and structures differed enough that classification required the creation of an entirely new family. At that time, only early life stages were available, leaving the adult form unobserved. That changed in 2001, when remotely operated vehicles captured images in the Gulf of Mexico that presented a fully developed individual. The footage revealed a body with an unusual configuration. Along each arm and tentacle, there is a distinct bend, forming a right angle that interrupts the otherwise continuous length. Beyond this bend, the limbs extend downward in straight lines, hanging beneath the body in long, vertical strands. The scale of these extensions becomes apparent when measured relative to the mantle. In observed individuals, the sections below the bend reach lengths 15 to 20 times the mantle length. For an animal with a mantle measuring around 30 centimeters, this produces appendages that extend several meters downward, forming a structure that occupies a large volume of the surrounding water. Movement is minimal. The body remains largely stationary, with only slight adjustments made through the slow undulation of its fins. These fins extend along much of the mantle, larger than those seen in most related species, and their motion is steady and controlled. The effect is one of suspension, maintaining position without visible effort. This posture is maintained at considerable depths, between 1,300 and 4,735 meters, within regions where observation is limited and encounters are rare. The arms remain extended below the body, not actively grasping or striking, but trailing through the water. The function of this configuration has not been directly observed. One proposed interpretation suggests that the extended arms act as a passive capture system, intercepting small organisms that come into contact with them. The limbs would serve as a kind of suspended array, increasing the area through which potential prey might pass. However, this remains unconfirmed, as no recorded footage has shown feeding behavior in detail. Encounters with Magnapinna have occurred across multiple ocean basins, including the Pacific, Atlantic, and Indian Oceans. This distribution indicates a wide presence, suggesting that the species is not restricted to a single region, but exists throughout deep marine environments globally. Despite this apparent range, physical specimens of adult individuals have not been collected. All available information about their mature form comes from video recordings and the interpretation of those images. The number of confirmed encounters remains low, fewer than a dozen observations documented over several decades of deep-sea exploration. The absence of specimens limits the ability to examine internal structures or confirm aspects of their biology that cannot be inferred visually. What is known is based on repeated patterns in the footage, consistent features that define the genus despite the scarcity of direct study. A body suspended in deep water, arms extending far beyond its central form, held in a position that suggests purpose without revealing it fully. The shape remains consistent across observations, yet its function continues to exist just beyond direct confirmation, present in view but not fully resolved. Light in this part of the ocean is not borrowed from above. It is made, released in brief signals that appear and vanish without leaving a trace behind. At depths beyond 500 meters, most living forms carry this ability, not as an exception but as a common feature, a shared capability woven into daily existence. Among squids, this capacity did not emerge once and spread. It appeared repeatedly in separate branches of their lineage, each arrival shaped independently by the same conditions. The result is not a single system but many, variations built from different biochemical pathways that arrive at a similar outcome, the controlled emission of light within a world that does not receive it from elsewhere. Some species produce light entirely within their own tissues. Chemical compounds are synthesized internally, held within specialized cells, and triggered into reaction when needed. The glow that follows is generated by the organism itself, a direct conversion of stored energy into visible form. Others rely on partnerships. Within certain squids, colonies of luminous bacteria reside inside dedicated organs, sustained and regulated by the host. These microorganisms emit light continuously, and the squid shapes that output through layers of tissue that act as shutters or lenses. The relationship is maintained through precise cycles. In one coastal relative, each morning brings a release, most of the bacterial population expelled into the surrounding water, leaving a small fraction behind. That remaining portion multiplies through the day, restoring the colony by night, aligning its brightness with the animal's period of activity. There are also species that do not produce or host light directly, but gather it through feeding. By consuming organisms that already carry light-producing molecules, they accumulate the necessary compounds and redirect them into their own structures. In this way, the capacity is acquired, transferred along a chain of predation and retention. The light itself is not uniform. It can be modulated, shaped, directed outward or downward, intensified or dimmed with precision. Some squids adjust brightness through reflective tissues that function like adjustable apertures, regulating how much light escapes into the water. The effect is controlled, not constant, tuned to match the surrounding conditions or to alter them. In most cases, the wavelengths produced fall within the blue and green spectrum, the range that travels furthest through seawater. Yet there are exceptions. A small number of species emit red light, a color that does not carry well in deep water and remains undetected by the visual systems of most other organisms. To those nearby, this illumination is effectively invisible. It allows the squid to reveal its surroundings while remaining concealed, a private field of vision that exists without announcing itself. There are also forms where intensity becomes the defining feature. In one species, large light organs positioned at the ends of its limbs produce flashes of remarkable brightness, brief but powerful, measured as some of the strongest biological emissions recorded in the ocean. These bursts are not continuous displays but controlled events, appearing in sequences that suggest timing and intent. Across these variations, light functions as more than a single-purpose tool. It operates as a medium through which information moves, a way to signal, to conceal, to reveal, to coordinate. Patterns are formed, pulses arranged, intervals maintained with consistency that implies recognition between individuals. In a space where sunlight does not reach, illumination becomes something that is generated, exchanged, and interpreted. It moves through the water in brief currents of brightness, forming a system that exists entirely within darkness, sustained by the organisms that produce it. There are moments in the deep when brightness does not arrive gently but in abrupt, concentrated bursts, as if the darkness itself has been briefly pierced. In those instants, the source is not distant or diffuse. It is close, deliberate, and moving with purpose. Taningia danae exists within that narrow interval between stillness and sudden motion. Its body carries a solidity uncommon among many of its relatives, compact and dense, shaped less for drifting than for directed movement. Measuring around 1.6 meters in length, it moves through the water with a controlled steadiness that suggests stored power rather than constant effort. Unlike most squid, it no longer possesses the pair of elongated feeding tentacles that define the group. Over the course of its lineage, those structures were lost, leaving 8 arms to perform every task. These arms are not reduced substitutes but specialized tools, each lined with hooks that curve inward, designed to secure rather than merely hold. When they close, they do so with finality. At the ends of 2 of these arms sit structures unlike anything else in the ocean. These are photophores, light-producing organs of unusual scale, positioned outward as if meant to face whatever approaches. Their brightness is not constant. It is stored, then released in short, intense emissions that illuminate the immediate surroundings for a fraction of a second before vanishing again. For many years, The presence of these organs was known only from preserved specimens, their purpose uncertain. The behavior of the living animal remained beyond observation, hidden within depths that resisted direct study. That absence persisted until a sequence of images, captured in the early years of the 21st century, offered a brief view into how this squid interacts with its environment. In that footage, the animal does not hesitate. It advances toward the source of disturbance, closing distance with a directness that contrasts with the slower movements seen in other species. Just before contact, the photophores activate. Light is released in rapid pulses, each flash following the last in quick succession. The effect is immediate, a sudden illumination within a space unaccustomed to it. The working interpretation of this sequence is grounded in timing. The flashes occur at the precise moment when proximity is greatest, when any nearby organism would be most affected by the abrupt change in light. In conditions where vision is adapted to low illumination, such intensity can disrupt perception, creating a brief interval in which movement is no longer accurately tracked. Within that interval, the arms extend and close. These same flashes have also been observed in interactions that do not lead directly to capture. When two individuals occupy the same space, similar bursts of light are produced, though with variations in rhythm and duration. The pattern suggests that the light serves more than one function, carrying signals that are received and interpreted by others of its kind. The absence of feeding tentacles has not limited its capacity to hunt. Instead, it has refined the sequence, bringing light and motion into closer alignment. The arms act as both reach and restraint, while the photophores shape the moment in which they are used. Evidence of its presence extends beyond direct observation. Its beak, dense and resistant to digestion, appears among remains recovered from predators that feed at depth. These fragments indicate that despite its size and capability, it is part of a larger exchange, one participant within a system that extends beyond any single encounter. Recorded across a range of depths, from several hundred meters downward, it occupies a span that intersects with multiple layers of the water column. Within that span, it carries a method that relies not on concealment or pursuit alone, but on altering the conditions of perception itself. In the absence of external light, it generates its own, not as a constant glow, but as a precise event, timed and directed, shaping the outcome of an interaction in a space where visibility is otherwise limited. Far below the surface, movement can be measured not by sight, but by pressure waves that travel through water, faint disturbances that expand outward and fade. In those depths, one of the largest predators on Earth descends regularly, following gradients that lead away from light and into regions where few other mammals can remain for long. The sperm whale moves through these layers with a physiology built for endurance. Its dives extend beyond 2,000 meters, with tracked descents suggesting even greater depths reached in routine patterns. Most of its existence unfolds in this vertical range, where it spends the majority of its time navigating cold, compressed water. Within that environment, its diet is dominated by squid, forming the bulk of its intake by weight. The scale of that consumption is substantial. A single adult requires hundreds of kilograms of squid each day to sustain its mass and metabolism. Across the global population, the cumulative intake exceeds that of all human fishing activity combined, measured not in isolated events, but in continuous, ongoing exchange between predator and prey. Yet the encounters that sustain this relationship remain unobserved. No direct recording has captured the moment when a whale meets a large squid at depth. Instead, the evidence arrives later, carried upward on the body of the whale itself. The skin holds marks that do not fade quickly. Circular impressions, evenly spaced, correspond to the gripping structures of one group of squid. Alongside them are linear patterns, deeper and more angular, produced by rotating hooks from another. These scars vary in size and arrangement, each one preserving the geometry of contact between two animals that met in darkness. Some markings extend beyond known dimensions. Rings far larger than those measured from any documented species appear across the whale's surface, suggesting interactions with forms that have not been directly examined. The skin becomes a record not only of known encounters but of those that remain outside classification. Additional traces accumulate within the digestive system. Squid beaks, resistant to breakdown, collect over time, forming dense clusters that reflect repeated feeding events. Around these structures, the whale's body produces a substance that encases and smooths them, gradually creating masses that can persist long after formation. When released into the ocean, these masses carry within them a compact archive, each beak representing a single animal taken at depth. From these fragments, patterns emerge. The shapes and sizes of beaks correspond to different species, allowing reconstruction of diet without direct observation. In some cases, the beaks do not match any known category, their proportions indicating animals that have not been otherwise recorded. The archive expands with each specimen examined, extending the boundary of what is recognized. The conditions under which these interactions occur remain constant: depths exceeding 1,000 meters, temperatures near freezing, pressures that compress and constrain. Within this setting, both whale and squid operate without light, relying on senses adapted to absence rather than presence. The events themselves unfold in silence, their details unrecorded, their sequences inferred only through aftermath. Movement, contact, resistance, and capture occur within a space that resists direct access. What remains are the impressions left behind, marks that indicate force and form but not motion. Through these traces, a picture forms, incomplete yet consistent. Predation is not a distant concept but an ongoing process, repeated across oceans, across years, occurring at depths that remain largely unobserved. The whale descends, the squid responds, and the interaction concludes without witness. The surface receives only the evidence, carried upward on skin and within tissue, quiet confirmations of encounters that continue beyond view, part of a system that operates regardless of observation, sustained by repetition and recorded only in fragments. At the smallest scale, far beneath muscle and motion, survival is decided in structures too small to see, arrangements of molecules that must remain stable in conditions that resist stability. Here, life does not depend on strength or speed, but on whether its chemistry can continue to function while compressed and chilled beyond ordinary limits. Every cell is enclosed by a membrane formed from phospholipids, molecules that align into a flexible boundary, allowing exchange, communication, and movement. In warmer, less pressurized water, this boundary remains fluid, shifting and adapting as needed. In the deep, the same structure faces opposing forces. Low temperature slows molecular motion, while pressure forces components closer together, reducing flexibility. Left unchanged, the membrane would stiffen, losing its capacity to function, and the processes it supports would cease. Deep-sea squids resolve this not by altering behavior, but by altering composition. Within their membranes, specific fatty acids are incorporated that do not align in straight, compact arrangements. Their shapes introduce subtle bends, preventing tight packing even under compression. The result is a boundary that retains its fluid character at temperatures near 2 degrees Celsius and pressures approaching 200 atmospheres, conditions that would immobilize less adapted systems. This adjustment is present in every cell, not as a temporary response, but as a fixed property of the organism. It is encoded, maintained across generations, and expressed uniformly throughout the body, ensuring that function persists regardless of depth. The same principle extends to proteins, the structures that carry out nearly every cellular task. In environments closer to the surface, these molecules fold into precise configurations, their shapes determining their function. Under increased pressure, that folding becomes unstable. The structures can distort or unravel, disrupting the processes they support. In deep-sea squid, the composition of these proteins differs in subtle but critical ways. Specific amino acids occupy positions that reinforce structural integrity, allowing the molecule to maintain its shape despite the surrounding force. Alongside these structural changes, chemical stabilizers accumulate within tissues. One such compound, trimethylamine oxide, is present in increasing concentrations as depth increases. It interacts with proteins at the molecular level, supporting their folded state and counteracting the effects of pressure. The relationship between its concentration and the depth at which a species lives is consistent enough that it can be used to estimate habitat, a chemical gradient reflecting the environment in which the organism operates. Buoyancy presents another constraint. Gas-filled structures, effective in shallower regions, compress under pressure, losing volume and function. In response, deep-sea squids rely on liquids that remain stable regardless of depth. Solutions rich in ammonium compounds fill internal spaces, their density lower than the surrounding seawater. This creates a balance, allowing the animal to maintain position without continuous movement, conserving energy in an environment where efficiency is essential. These features are not isolated adaptations, but parts of a coordinated system. Membranes remain flexible, proteins retain their form, internal chemistry supports stability, and body composition maintains equilibrium within the water column. Each element addresses a different aspect of the same challenge, ensuring that the organism functions as a whole. The process by which these traits emerged extends across vast spans of time. Variations that failed to operate under pressure were removed, while those that maintained function persisted and accumulated. The result is a form of life whose fundamental operations are tuned to conditions that differ profoundly from those near the surface. In this way, survival is carried not only in visible form, but in molecular arrangement, a pattern established at scales too small to observe directly, yet essential to every movement and every moment of existence in the deep. In certain stretches of the eastern Pacific, the water itself seems to move with a collective rhythm, not a current but a shifting presence, as if many bodies are advancing together just beyond clear sight. Within that movement, individual boundaries blur, replaced by a coordinated flow that changes direction without scattering. Dosidicus gigas occupies this space as both participant and organizer. Reaching lengths of around 1.5 meters and masses near 50 kilograms, it carries a strength that allows it to move with purpose through varying depths. Its history is tied to the cold upwelling of the Humboldt Current, where nutrient-rich waters once defined the limits of its distribution. Unlike many squid that operate alone, this species gathers in large groups. These aggregations are not passive clusters but structured formations, within which individuals maintain spacing and alignment. During feeding, this organization becomes more apparent. Movements are not random. They occur in sequences, with individuals advancing and retreating in patterns that suggest coordination rather than coincidence. Observations made in controlled studies have revealed flashes of color moving across their bodies in rapid succession. These changes, shifting from deep red to pale tones in fractions of a second, pass from one individual to another in ordered intervals. The signals are brief yet consistent, forming sequences that appear to guide collective behavior. Analysis of these patterns indicates that information is being transmitted, though the exact nature of that information remains unresolved. Within this coordinated activity, hierarchy is not fixed. Any individual that becomes injured or slows its movement risks becoming part of the next feeding event. Cannibalism is not an anomaly, but an integrated aspect of their behavior, a mechanism that ensures energy remains within the group. The boundary between participant and prey can shift quickly, depending on condition and position. Their growth reflects a different kind of urgency. From egg to full size, the entire lifespan unfolds in approximately 1 year. Development is rapid, driven by constant feeding and efficient conversion of energy. There is no extended period of maturity. Instead, the cycle moves from emergence to reproduction and then to death within a single sequence of seasons. Daily movement extends vertically through the water column. At night, they rise toward shallower depths where food is more abundant. As daylight returns, they descend into layers where oxygen levels are low, regions that many fish cannot tolerate. Within these zones, their physiology allows continued function, granting them access to areas that serve as refuge from larger predators. In recent decades, their distribution has expanded. Sightings now occur far beyond their historical range, extending along coastlines that were once outside their regular presence. This shift corresponds with broader changes in ocean conditions. As temperatures rise and competing species decline, new regions become accessible, and the squid occupy them with notable speed. Their expansion is not gradual. It occurs in steps that reflect both opportunity and adaptability. Where resources become available and resistance decreases, they establish themselves quickly, forming new aggregations that mirror those of their original habitat. Throughout this process, communication remains central. The rapid color changes observed during feeding likely serve additional roles, maintaining cohesion as groups navigate shifting environments. The signals persist across contexts, suggesting a flexible system that supports both coordination and response. In these waters, they function as both hunters and participants in a larger transformation. Their presence alters local dynamics, influencing prey populations and interacting with existing species in new ways. As conditions continue to change, their range and behavior adjust accordingly, maintaining a balance that is responsive rather than fixed. The movement of these groups through the ocean reflects a form of collective adaptation shaped by immediate conditions and carried forward through each generation that completes its brief, accelerated life. Before a squid moves, before it changes direction, or adjusts its position, something else shifts first, a transformation that begins across the surface of its body. Color spreads, contracts, and reorganizes in patterns that seem less like decoration and more like an active process unfolding in real time. At the level of a single square millimeter, this surface is densely structured. Hundreds of chromatophores are embedded within the skin, each one a minute sac of pigment surrounded by a ring of muscle fibers. These fibers connect directly to the nervous system, forming a link between intention and appearance. When they contract, the pigment sac expands outward, changing from a nearly invisible point into a disc that can be seen from a distance. The expansion occurs rapidly, increasing visible area hundreds of times within a fraction of a second. Each chromatophore operates as an independent unit. A dedicated motor neuron controls its state, allowing precise activation or relaxation. This arrangement creates a distributed field of control where the brain can address each element individually rather than in large groups. Across the body of a large squid, this results in tens of thousands of controllable points, each capable of shifting in coordination or isolation. Beneath this layer, additional structures contribute to the overall effect. Iridophores lie below the pigment cells, producing color not through pigments, but through the interaction of light with microscopic layers. Their output changes depending on angle, creating shifting tones that respond to movement and position. These cells do not require direct neural input in the same way, instead altering appearance through physical properties that remain constant. Further below, leucophores form a base layer that reflects and scatters incoming light. This diffuse reflection provides a neutral background, enhancing the visibility of the upper layers and allowing combinations of effects that would not be possible with pigment alone. Together, these three layers form a composite system, each contributing a different aspect of the visible result. The combined effect is a surface capable of rapid transformation. Patterns emerge and dissolve in less than 200 milliseconds, even as the animal continues to move. Shapes can be defined, edges sharpened or softened, textures suggested through subtle variations in tone. The body becomes a dynamic field, continuously adjusting in response to internal and external signals. This capacity extends beyond simple changes in color. Some squid generate patterns that travel across their bodies, bands of contrast that move in coordinated sequences. These patterns are not random fluctuations, but structured events, appearing with consistency and repeating in recognizable forms. When multiple individuals are present, similar sequences can pass between them, suggesting a form of exchange that relies on visibility rather than sound. The system is flexible enough to support simultaneous functions. One side of the body can present a pattern distinct from the other, allowing different interactions to occur at once. This division is controlled and maintained without visible delay, indicating a level of coordination that integrates sensory input with motor output across the entire surface. The neural demand required to sustain this activity is substantial. Processing must occur continuously, translating perception into immediate change. Despite the relatively compact size of the central brain, the distribution of neurons throughout the body supports this load, extending processing capacity into the arms and skin itself. In this way, the surface is not a passive covering, but an active interface, responding, signaling, and adapting in a continuous flow. It carries information outward, while also reflecting internal states. A system where appearance is directly tied to function, unfolding across the body in patterns that exist only for the moment they are needed. There are distances in the ocean where an individual may pass through an entire life without encountering another of its kind. The water stretches in all directions, unmarked and without reference, and within it each body moves alone, guided only by faint signals that dissipate almost as quickly as they form. For species living in this expanse, reproduction is not a matter of timing or display, but of chance, compressed into a single moment. When two individuals meet, the opportunity exists only briefly, and whatever decision is made must be immediate, without certainty or conformation. In the mid-water layers of the Pacific, one squid has resolved this uncertainty through a method that removes hesitation entirely. Octopotuthis deletron encounters others of its species in near darkness, where visual distinction offers little guidance. Rather than attempting to identify sex before mating, the male proceeds without discrimination, treating every encounter as a potential success. The mechanism differs from the more familiar transfer of spermatophores through a specialized arm. Instead, the male uses an extensible organ to implant packets of sperm directly into the tissue of the other individual. These packets embed themselves beneath the surface, remaining viable within the body until conditions allow fertilization. The act is brief, direct, and does not depend on recognition. Examination of specimens has revealed a pattern that confirms the consistency of this approach. Both males and females carry embedded spermatophores in similar numbers, distributed across the body without clear distinction. This symmetry reflects a system in which the risk of error is accepted as part of the process, outweighed by the necessity of acting when contact occurs. In such an environment, missed opportunities carry a greater cost than misplaced effort. The energy invested in producing sperm is significant, yet the probability of encountering another individual is so low that withholding action would reduce reproductive success more severely than any inefficiency. Over time, this balance has shaped behavior toward immediacy rather than selectivity. Elsewhere in the deep, a different solution unfolds through persistence rather than speed. The female Gonotis onyx gathers her eggs into a single mass, holding them within her arms as they develop. This mass remains with her continuously, suspended in the surrounding water, its surface gently moving as she circulates currents through it. For months, she maintains this position. Feeding ceases, and her reserves diminish gradually as the embryos grow. The duration extends across seasons, each day marked by the same slow exchange between her body and the developing cluster she carries. Movement becomes minimal, limited to what is necessary to sustain the eggs. As the embryos approach maturity, her condition declines. The energy required to maintain both her own tissues and the growing mass can no longer be met. When the young finally emerge, she has reached the limit of what her body can provide. Her role concludes as theirs begins. The transfer complete. Other species release their offspring into the open water without protection. Their egg masses form large, gelatinous spheres that drift freely, containing vast numbers of developing embryos within a transparent structure. These spheres move with currents, unguarded, relying on scale rather than defense to ensure that some will survive. Across these variations, a common pattern remains. Reproduction is shaped by rarity, by the low frequency of encounters, and the absence of reliable signals. Strategies diverge in form but converge in purpose, each one adapting to a world where continuity depends on actions taken in isolation, guided by probabilities rather than certainty, carried out in a space where even the presence of another is never assured. Within a body that rarely pauses, signals move along pathways arranged in ways that do not mirror the familiar designs of vertebrate life. The organization is distributed, layered, and partially decentralized, forming a system where control is shared rather than held in a single place. The brain is large for an invertebrate, yet it is only one center among many. A squid carries approximately 500 million neurons, a number that places it among the most neurologically complex animals outside the vertebrates. What distinguishes this system is not only its size but its arrangement. Nearly 2/3 of these neurons reside in the arms, embedded within dense nerve cords that extend the length of each limb. These networks process information locally, allowing the arms to respond to contact chemical signals, and texture without waiting for instructions from the central brain. An arm can explore a surface with a degree of independence, adjusting its movements as it encounters variations, coordinating suckers in sequences that adapt in real time. This creates a form of parallel processing where multiple parts of the body gather and interpret information simultaneously. The central brain does not dictate every action, but integrates outcomes, shaping broader patterns of behavior from many smaller decisions occurring at once. Laboratory observations, limited though they are, have revealed capacities that extend beyond simple reflex. Squids can learn associations between neutral cues and consequences, altering their responses over repeated exposures. The changes persist, indicating that memory is not transient, but stored in a way that influences future behavior across time. More intricate patterns appear when context is involved. Experiments suggest that squids can retain information not only about an event, but about its surrounding conditions, including location and sequence. This form of episodic-like memory allows them to navigate situations with a sense of prior experience, adjusting actions based on what has previously occurred in similar circumstances. When presented with unfamiliar objects that do not resemble food or threat, their behavior does not default to avoidance or indifference. Instead, they approach, examine, and manipulate, using arms and suckers to gather information. The movements are measured, varied, and exploratory, as if testing possibilities rather than reacting to a predefined category. Communication adds another layer to this system. The dynamic patterns expressed across the skin require continuous interpretation of signals from others, followed by rapid generation of responses. These exchanges unfold in fractions of a second, demanding coordination between perception, decision, and execution at a pace that leaves little room for delay. The neural load involved in sustaining such interactions is substantial, drawing on both centralized processing and the distributed networks within the body. Studying these capacities presents persistent challenges. Many species live for only a short span, compressing development, learning, and reproduction into a limited period. Maintaining them in controlled environments proves difficult, as their physiology responds poorly to confinement and altered conditions. Observations often capture fragments rather than complete behavioral arcs. For species that inhabit greater depths, the difficulty increases further. Changes in pressure during ascent alter their bodies in ways that disrupt normal function, leaving little opportunity to observe natural behavior. The environments in which their cognition operates remain largely inaccessible, beyond the reach of sustained human presence. Intelligence in this context unfolds within isolation, shaped by brief encounters and the absence of stable social groups. It is expressed through interaction with a world that offers few consistent cues, requiring flexibility and rapid adjustment. The processes that support this way of living remain only partially visible, glimpsed through experiments and fleeting observations, suggesting a form of awareness that follows pathways still unfamiliar to human understanding. Long after the surface settles into darkness, movement begins far below, not as a single wave but as countless individual ascents unfolding together. Bodies rise through the water column in a slow, coordinated expansion, drawn upward by the availability of energy concentrated near the light above. By the time night fully settles, entire layers of the ocean have shifted position, lifted hundreds of meters closer to the surface. This migration occurs every night across every ocean basin, involving a mass of life that exceeds any other regular movement on the planet. The scale is difficult to hold in a single image, measured in hundreds of millions of tons traveling vertically, then returning before the first trace of daylight restores visibility to the upper water. It is a rhythm older than most modern ecosystems, repeating without interruption across vast spans of time. Among those moving within this cycle are squids, including species such as Beryteuthis magister, whose journeys trace a path between midwater depths and the upper layers. Over the course of a single night, they may travel from several hundred meters below to regions where food is abundant, then descend again before exposure increases. The motion is deliberate, conserving energy while navigating gradients of temperature and pressure that shift with depth. At the surface, microscopic organisms have already transformed sunlight into organic matter through photosynthesis. This process fixes carbon from the atmosphere into living tissue, creating a resource that supports the entire marine food web. When squids feed in these upper layers, they incorporate that carbon into their own bodies, converting it into muscle, tissue, and energy reserves that will travel with them as they descend. What follows is a transfer that extends beyond immediate survival. As these animals move downward, they carry with them the material formed near the surface, relocating it into deeper regions where it would not otherwise accumulate. Waste products produced during this descent, along with uneaten fragments and shed material, begin a gradual fall through the water, settling into layers where decomposition slows. When a squid completes its life cycle and its body sinks, the process continues. Carbon that once circulated near the surface becomes part of the deep ocean, isolated from the atmosphere for extended periods. Predators that consume these animals contribute in a similar way as their own remains eventually descend, adding to the downward movement of organic matter. This continuous transfer is known as the biological pump, a system that moves between 5 and 12 gigatons of carbon each year from the upper ocean into depths where it can remain stored for centuries or longer. The effect is cumulative, sustained by the combined actions of countless organisms participating in daily migration and life cycles that conclude far from where they began. Without this movement, carbon would remain closer to the surface, maintaining a stronger connection with the atmosphere. Concentrations of carbon dioxide would rise accordingly, altering conditions in ways that extend far beyond the ocean itself. The stability of the climate is linked, in part, to the quiet persistence of this exchange. Squids occupy a place within this system not as a singular driver, but as one of its active components. Their movements, feeding, and eventual descent contribute to the larger pattern, integrating them into a process that operates continuously across the planet. Within the darkness below, this exchange proceeds without interruption, shaping conditions far beyond the depths where it occurs. Between the illuminated surface and the deeper layers below, there exists a band of water where life thins, not from absence of nutrients, but from the quiet depletion of breath itself. In these midwater depths, oxygen has been steadily consumed, drawn down by the slow work of bacteria, breaking apart the constant descent of organic matter from above. What remains is a region where survival depends not on finding food, but on enduring scarcity at the most fundamental level. These zones form between roughly 200 and 1,000 meters, their boundaries shifting with currents and temperature. Within them, dissolved oxygen falls to levels that most marine animals cannot tolerate. Fish that pass through begin to falter quickly, their metabolism demanding more than the environment can provide. The water becomes a barrier, an invisible boundary that reshapes movement and distribution throughout the ocean. Yet, for certain squids, this same region functions not as a boundary, but as a refuge. Their physiology allows them to occupy spaces that exclude competitors and predators. turning limitation into advantage. The conditions that restrict others create a form of protection, reducing encounters and opening territory that would otherwise be contested. In species adapted to these layers, oxygen extraction operates at the edge of what chemistry allows. The copper-based hemocyanin in their blood binds oxygen with an affinity tuned to these depleted conditions, enabling uptake even when concentrations fall to a small fraction of those near the surface. Circulation and respiration are adjusted to maximize efficiency, maintaining function where most systems would fail. Some species extend this adaptation further by altering their metabolic demands. As they enter these regions, their energy use declines in a controlled manner, reducing the amount of oxygen required to sustain basic processes. Movement slows, activity becomes measured, and physiological systems shift into a state that balances minimal intake with essential function. This capacity allows them to pass through and remain within layers that would otherwise be inaccessible. Over recent decades, these oxygen minimum zones have expanded. Measurements from long-term monitoring and satellite observations indicate an increase in their total area by several million square kilometers since the mid-20th century. Warmer ocean temperatures reduce the solubility of oxygen in seawater, while enhanced biological activity increases consumption, deepening and widening these regions across multiple basins. As these zones grow, the structure of marine ecosystems adjusts in response. Habitats suitable for oxygen-sensitive species contract, limiting their range and concentrating populations into narrower bands. At the same time, species capable of tolerating low oxygen encounter fewer barriers, extending their presence into newly formed regions. Squids adapted to these conditions are among those expanding their reach. Their ability to function within low oxygen environments allows them to occupy spaces that others abandon, altering interactions within the food web. Predators that depend on higher oxygen levels are restricted, while prey species may shift their distributions in response to changing pressures. This transformation unfolds gradually, measured through long-term data rather than immediate observation, yet its effects accumulate. The balance between different groups of organisms changes as the environment redefines what is possible for each. Squids do not initiate this shift, yet their biology aligns closely with the new conditions, allowing them to persist and expand as others retreat. Within these dim layers, the distribution of life continues to reorganize, guided by the subtle but persistent influence of oxygen availability. The ocean reshapes itself through processes that operate beyond immediate perception, and within that reshaping, certain forms find new space to endure. Long before cameras reached into the depths, the first attempts to understand what lived below relied on contact rather than observation. In the late 19th century, the HMS Challenger expedition lowered nets into waters that had never been sampled, drawing them back with fragments of life that confirmed what had only been suspected. The deep ocean was not empty, but inhabited, though what those inhabitants were like remained largely unknown. The method was simple in concept and limited in precision. Nets descended through layers of water, open as they fell, gathering organisms indiscriminately before closing and returning to the surface. What arrived aboard carried the combined imprint of multiple depths, making it difficult to determine where any individual had originated. By the time these specimens emerged, the transition from pressure and cold to surface conditions had altered or destroyed much of what made them representative of their environment. Refinements followed, including mechanisms designed to close nets at specific depths, isolating samples more effectively. Even so, the process remained extractive. The organisms collected were no longer behaving as they had below, and delicate forms often failed to survive the journey upward. Information could be gathered about structure, but not about movement, interaction, or the subtle rhythms of life in place. A different approach emerged with the development of remotely operated vehicles. These machines descended with cameras and lights, transmitting images from within the environment itself. For the first time, animals could be observed alive, moving through their natural surroundings without the distortions introduced by retrieval. Behaviors that had never been recorded began to appear, captured not as preserved remains but as unfolding actions. Light, however, presented its own complications. Many deep-sea organisms respond to illumination in ways that alter their behavior, making observation intrusive. To address this, systems were developed that operate outside the visual range of most species. Among them, the Medusa camera system employs far-red light, effectively invisible in the deep, paired with a lure that imitates a specific bioluminescent signal associated with distress in certain gelatinous organisms. This combination attracts attention without triggering avoidance, allowing encounters to occur with minimal disturbance. Through such methods, encounters have been recorded that extend beyond what earlier techniques could achieve. A giant squid, once known only from indirect evidence, was filmed in these conditions within the Gulf of Mexico, appearing as it would in its own environment rather than as a specimen altered by retrieval. Alongside visual observation, other approaches have expanded the ability to detect presence without direct contact. Environmental DNA sampling analyzes fragments of genetic material suspended in seawater, released through skin cells, mucus, or waste. By filtering and sequencing this material, researchers can identify species that have passed through an area even when no individual has been seen. Acoustic techniques provide another layer of information. Variations in density within the water column, including those produced by internal structures such as the gladius, reflect sound waves in detectable ways. By interpreting these reflections, it becomes possible to estimate the distribution and abundance of certain organisms across large areas without capturing them. Despite these advances, the scope of observation remains limited. The cumulative time spent exploring the deep ocean with such technologies represents only a small fraction of the available habitat. Each descent, each recording, samples a narrow path through an immense volume, offering glimpses rather than comprehensive views. What is known is shaped by where instruments have been placed and when they have been deployed. Beyond those points, vast regions remain unobserved, their inhabitants continuing without record. The act of looking has progressed from retrieval to presence, yet the scale of what remains beyond reach continues to define the limits of understanding. Within the soft architecture of a squid, one structure resists change even after death. The beak, composed of chitin, remains intact when everything else dissolves, holding its form long after muscle and tissue have been broken down. It is dense, sharply defined, and nearly impervious to digestion, a fragment designed for function that becomes, in time, a record. Inside the stomach of a sperm whale, these beaks accumulate. Each feeding dive ends with soft matter reduced to energy, while the beaks persist, gathering slowly over years into a layered archive. They settle together, forming a collection shaped not by intention but by repetition, each one marking a separate encounter in water far below the surface. When these collections are examined, patterns emerge alongside irregularities. Many beaks correspond to species already described, their shapes matching known forms cataloged through previous study. Yet among them are others that do not align with any existing classification. Their proportions differ, their curvature or structure falling outside the range defined by known species. These differences are not subtle variations within a familiar group. In some cases, the distinctions are pronounced enough to indicate entirely separate lineages. Researchers studying these collections have noted that a consistent portion of the beaks cannot be assigned to any documented species, suggesting the presence of organisms that have not yet been observed directly. The size of certain beaks adds another dimension to this absence. Measurements taken from their structure allow estimates of the animals that produce them. By analyzing specific features, such as the lower rostral length, scientists can infer total body mass using established relationships derived from known species. Some of these unidentified beaks correspond to sizes that exceed the largest confirmed specimens currently recognized. This implies the existence of animals of considerable scale, moving through depths where observation remains limited. Their presence is inferred not from sight or capture, but from the remains carried within predators that traverse those regions. The whale becomes an intermediary, its feeding behavior sampling environments that remain otherwise inaccessible. Long-term studies have expanded these observations beyond a single source. Collections assembled over decades include specimens from multiple predators, each contributing a different perspective based on the depths and regions they inhabit. Sleeper sharks, elephant seals, and albatrosses each carry fragments of encounters, their stomach contents reflecting the diversity of life encountered across different layers of the ocean. Across these varied sources, the pattern remains consistent. There are forms present in the deep that have not been documented through direct observation. Their beaks appear in collections, distinct and recurring, yet the animals themselves remain unseen, their behaviors and full anatomy unknown. Researchers who have devoted years to cataloging these structures, comparing shapes and measurements across thousands of samples, continue to encounter forms that resist classification. Each unidentified specimen adds to a growing indication that the known diversity of squid represents only a portion of what exists. The evidence is indirect, assembled from fragments rather than complete organisms, yet it is persistent. It suggests a population of species that move through the deep without entering the narrow paths where cameras or nets have been deployed. Their presence is confirmed through what remains after predation, a record preserved within the bodies of those that have encountered them. In this way, the deep ocean reveals itself not through direct visibility but through traces carried upward, fragments that point toward forms still beyond reach. In a quiet storage room, far from the environment it once occupied, a single preserved form rests within a sealed container. It is catalogued, measured, and labeled, its presence reduced to a record that can be revisited but not expanded upon. There is no movement to observe, no behavior to interpret, only the fixed outline of a body that arrived from depth and never returned. Chromatophores sulcus is known in this way, through one individual collected from the South Atlantic at a depth of approximately 2,000 meters in 2007. Its discovery did not begin with recognition of its rarity, but with confusion. Images of the specimen circulated widely, drawing attention to a feature that seemed almost familiar, a ring of structures around the beak that resembled human teeth. The resemblance was an artifact of preservation. The structures in question were not teeth, but labial folds, thickened tissue surrounding the beak. In life, they would have formed a different arrangement, but during preservation they collapsed and dried into a configuration that suggested something else entirely. The interpretation spread rapidly, repeating across networks without correction, shaping perception long before clarification reached the same audience. What remains after this misinterpretation is an organism that requires no embellishment. Its anatomy includes labial folds, more pronounced than those of related species, forming a structure whose function is not yet understood. With only a single specimen available, there is no basis for comparison, no way to determine whether this feature varies within the species or remains constant across individuals. The specimen itself is housed in the Natural History Museum in London, where it serves as the entire foundation for scientific understanding of this species. From it, measurements have been taken, features described, and a classification assigned. Beyond these details, the record does not extend. Its size range in life, its distribution across depth or geography, its diet, its reproductive cycle, and its behavior remain unobserved. This absence of information is not unusual. Across the study of deep-sea cephalopods, many species are represented by similarly limited material. Some are known from only one or two individuals, collected at specific times and places, with no subsequent encounters to refine or expand the description. Each specimen becomes both evidence and limitation, defining what is known while marking the boundaries of what remains uncertain. The environment from which these animals originate complicates further study. Retrieval alters their condition, and opportunities to observe them in place are rare. As a result, the gap between presence and understanding persists, with many species existing in scientific literature as names supported by minimal physical reference. For those species that have been observed alive, even briefly, the available knowledge extends into behavior and interaction. Yet, for many others, including Chromatophores sulcus, the narrative ends at the point of collection. There is no sequence of observations, no accumulation of detail over time, only the initial encounter preserved. Within this framework, the deep ocean appears not as a fully described system, but as a collection of partial records. Each specimen contributes a fragment, yet the broader picture remains incomplete, shaped by what has been encountered rather than by what is present. The single individual of Chromatophores sulcus stands within this context as both a discovery and an indication of absence. It confirms that such forms exist while leaving open nearly every question about how they live. Far below the reach of seasons, where change was once measured in geological patience, a different rhythm has begun to register. Instruments lowered into the abyss return not with silence, but with numbers that drift, small at first, then persistent. Temperatures at depths of 4,000 meters have risen in multiple ocean basins, subtle shifts carried through water that was long believed insulated from the atmosphere above. This stability had been assumed, not proven. The deep ocean, vast and layered, seemed too distant to respond within the span of human observation. Yet currents connect what appears separate, and heat, once introduced, finds pathways downward. The result is not a sudden transformation, but a gradual adjustment, measurable only through careful, repeated sampling over years that accumulate into decades. Alongside temperature, the chemistry of the water itself is changing. Carbon dioxide absorbed at the surface dissolves into seawater, forming carbonic acid that spreads through the ocean's interior. The alteration is slight in absolute terms, but precise systems respond to slight shifts. In squid, one of these systems lies within structures no larger than a grain of sand. Statoliths, composed of calcium-based material, serve as internal reference points for balance and motion. They allow the animal to sense orientation, to register acceleration, to maintain position in a three-dimensional space where there are no fixed landmarks. Their formation depends on the surrounding chemical environment, and when that environment shifts, so does their development. Laboratory studies that recreate projected future conditions show changes in these structures. Crystals form irregularly, surfaces become uneven, shapes diverge from the patterns observed under stable conditions. These differences suggest altered function, not through damage that can be seen from a distance, but through minute deviations in the geometry that underlies perception of movement and position. While some changes emerge within the body, others unfold across geography. The distribution of certain squid species has shifted in response to warming waters, extending into regions that were previously outside their tolerance. In the eastern Pacific, populations once concentrated along the cold upwelling of the Humboldt Current have moved northward by thousands of kilometers. Encounters in higher latitudes, including waters near Alaska, have been documented within recent decades. Such appearances would have been unlikely within living memory, constrained by temperature boundaries that have since shifted. As these limits move, so do the organisms adapted to follow them, filling spaces left by species less able to adjust. The pace of this adjustment is shaped by the nature of squid life histories. Many species live for 1 to 2 years, completing their entire cycle within a narrow window of time. Each generation represents a new iteration, subject to selection pressures that operate continuously. Changes in environment are therefore met not by individuals adapting over long lifespans, but by populations shifting across successive generations. This responsiveness allows for rapid adjustment in distribution and physiology, yet it also means that each generation is exposed directly to the conditions of its time. There is no buffering through longevity, no extended period during which previous adaptations can persist unchanged. The relationship between organism and environment becomes immediate, recalibrated with each cycle. In this way, the deep ocean, once considered constant, reveals itself as responsive, linked to processes occurring far above. Squids, present across these depths, continue to adjust within this shifting context, their forms and ranges reflecting the conditions through which they move. The transformation is ongoing, not defined by a single moment, but by accumulation. Within it, the outlines of familiar patterns begin to change, quietly, steadily, carried through water that remembers every alteration. In drawers and databases, new names continue to appear, quietly added to a catalog that never seems to approach completion. Since the turn of the millennium, between 20 and 30 squid species have been formally described every decade, each one recognized from fragments of evidence gathered in places where observation is brief and uncertain. The pace has not slowed. If anything, it has steadied into a rhythm that suggests not a nearing endpoint, but an expanding horizon. A coordinated effort at the beginning of this century, the Census of Marine Life, attempted to measure the scale of what remained unseen. After 10 years of sampling across oceans, depths, and habitats, the conclusion settled with a kind of understated gravity. Approximately 91% of marine species had yet to be described. For organisms living far below the reach of routine exploration, that fraction rises even higher, shaped by distance, pressure, and the fragility of the animals themselves. Traditional collection has always imposed limits. Nets distort delicate bodies. Pressure changes erase natural form, and retrieval transforms living structure into something altered beyond recognition. Increasingly, the act of discovery no longer depends on bringing an organism to the surface. Cameras descend instead, observing animals in place, capturing motion, posture, and interaction in ways that preserved specimens never could. From these recordings, taxonomists have begun to recognize species without ever holding them. Subtle differences in fin shape, arm arrangement, or the sequence of movements through water become distinguishing features, patterns consistent enough to suggest identity. In some cases, a species is known only from these moments, defined by appearance and behavior alone, existing in classification without a physical counterpart in any collection. Alongside vision, another method has emerged, one that leaves no image at all. Seawater carries traces of life in fragments too small to see, cells shed from skin, mucus, or waste. By filtering this water and analyzing the genetic material within it, researchers detect the presence of animals that have passed through unseen. A sequence of nucleotides becomes evidence, a signature of an organism that may never be observed directly, yet whose existence is confirmed by the chemistry it leaves behind. These approaches reveal patterns that extend beyond individual species. Signals appear in places where no specimen has ever been collected, suggesting communities that remain entirely outside direct observation. Each detection adds a point to a map that is still largely blank, hinting at structures and relationships that have yet to be resolved. Even among larger animals, absence persists. The Magnapinna, with arms extending far beyond its body, has been recorded fewer than a dozen times across decades of exploration. Its scale does not guarantee visibility, and its rarity in recorded encounters suggests that size alone does not determine discoverability. If such a form can remain so infrequently observed, then smaller or more cryptic species may pass through the same spaces without leaving any visual record at all. Occasionally, the ocean delivers a brief moment of clarity. A stranding along a coastline, a cluster of individuals brought together by conditions not fully understood, provides an opportunity to observe behavior directly. In one such event, along the coast of Chile, a species related to Dosidicus displayed patterns of movement and interaction not previously documented, requiring adjustments to what had been assumed about its life. Each year, new observations emerge from scattered efforts: a trawl, a descent, a sample drawn from water that appears empty. Some match what is already known, others do not. The distinction between familiar and unfamiliar remains fluid, shifting as each edition reshapes the boundaries of the catalog. This accumulation does not suggest completion. It suggests that the work has only begun, that the largest habitat on Earth continues to reveal itself in fragments, each one precise, each one incomplete, together forming a picture that is still in the process of becoming. It becomes possible, after a certain point, to read these bodies not only as organisms, but as records. Each form carries within it a history that was never written, only tested, again and again, in conditions that offered no tolerance for error. What remains is not a design, but a residue of survival, shaped by absence as much as presence. Every feature traced through these animals, from the molecular arrangements that keep cells fluid under crushing weight to the orientation of an eye that avoids casting a shadow, exists because alternatives did not persist. Nothing was granted in advance. Each solution emerged from sequences in which slight advantages accumulated, while less effective variations vanished without record. The ability of a species to remain active in water containing only a fraction of the oxygen found at the surface reflects this process with particular clarity. It is not a single adaptation, but the outcome of countless incremental differences, each one affecting the duration of survival by a margin too small to perceive in isolation. Over time, those margins compound, leaving behind a lineage capable of existing where most cannot. This pattern extends outward, visible in structures that appear almost excessive when viewed without context. Eyes that reach dimensions unmatched by any other animal for instance, are not expressions of abundance but of necessity refined across vast spans of time. Each slight increase in sensitivity or range would have altered the probability of detecting movement in darkness, shifting the balance between encounter and avoidance in ways that favored continuation. In another form, precision appears as alignment between structure and environment so exact that it resembles intention. The filtering of specific wavelengths of light matched to the spectral qualities present at a particular depth demonstrates a correspondence that is both narrow and exacting. Such correspondence arises not from foresight, but from repeated selection against anything less accurate. What becomes apparent is not a process driven toward complexity for its own sake, but one constrained by conditions that admit only certain outcomes. Variation exists continuously, yet only a fraction of that variation aligns with the demands imposed by pressure, temperature, and absence of light. Those that align remain. Those that do not leave, no continuation. In more forgiving environments, this process can appear diffuse, its effects softened by broader margins within which life can persist. Here, the margins narrow. The relationship between structure and survival sharpens, and the consequences of deviation become immediate. In that narrowing, the underlying mechanism becomes more legible, not because it changes, but because it is less obscured. These organisms do not represent exceptions to a general pattern. They reveal it more clearly. The same principles operate across all life, though often hidden beneath conditions that allow for greater variation without immediate consequence. In the deep, where constraints are strict and resources limited, the pattern resolves into forms that appear both unfamiliar and precise. There is a continuity that extends through all of it, connecting each present form to a sequence of prior states, each one tested in turn. What is visible now is the outcome of that sequence, not its entirety. The process continues, as it always has, without direction, without conclusion, proceeding through iterations that leave behind only those configurations that endure. Seen in this way, the deep ocean functions as a kind of reflection, not of intention, but of process itself. It reveals what remains when conditions remove all but the most consistent outcomes, leaving behind structures that embody the constraints under which they were formed. Seen from the surface, the ocean appears continuous, a single body of water stretching beyond the horizon. Yet far below that shifting boundary lies a domain whose persistence predates nearly everything familiar on land. For approximately 200 million years, the deep ocean has remained in a state remarkably similar to its present condition, a stable volume of darkness, pressure, and cold extending across the majority of the planet. During that span, continents have fractured and reassembled, mountain ranges have risen and worn down, climates have oscillated between extremes, and entire ecosystems have emerged and vanished. The deep, by contrast, has changed slowly, its defining characteristics maintained through cycles that altered the surface repeatedly. It is not static, but its variations occur on scales that make continuity the dominant impression. The organisms within this environment are not recent arrivals. They are part of lineages that extend back through that long continuity, each generation linked to those that came before without interruption. What is encountered today represents the current form of processes that have been unfolding since long before the appearance of mammals, carried forward through conditions that remained consistent enough to allow such persistence. When a specimen is brought upward or an image is captured in situ, the encounter is not merely spatial, it is temporal. The form observed has been shaped by pressures that have acted over durations difficult to represent in human terms, each structural detail reflecting responses to constraints that have been present for vast stretches of time. The result is an organism that embodies a history far longer than its individual lifespan. At depths approaching 1,000 meters, light from the surface no longer penetrates. This is not a temporary absence, but a permanent condition. No photon from the sun reaches these layers, not now and not at any point during the period in which the modern deep ocean has existed. The environment is defined by that absence, and the organisms within it have developed in relation to it. Adaptations in such conditions do not compensate for missing light. They incorporate its absence as a constant. Sensory systems, behaviors, and interactions are organized around a world in which illumination from above has never been a factor. The forms that result are not incomplete versions of surface organisms, but configurations that are coherent within their own context. Within this darkness, light appears, generated by the organisms themselves. Bioluminescence forms a network of signals, exchanges, and interactions that function independently of the sun. Each flash, pulse, or glow participates in a system that has been operating for durations that precede the earliest terrestrial mammals. It is a mode of communication and interaction that developed entirely within the deep, sustained by conditions that have remained largely unchanged. The continuity of this environment allows such systems to persist across immense spans of time. While surface ecosystems undergo rapid transformation, the deep maintains a framework within which long-term processes can unfold with fewer interruptions. The organisms encountered there are part of that continuity, not anomalies but expected outcomes of sustained conditions. What becomes visible through observation is not an isolated phenomenon, but a segment of an ongoing process. The deep ocean is not empty or transitional. It is a complete environment with its own dynamics, histories, and patterns of interaction, operating independently of the variability above. To look into it is to encounter a system that has been in operation far longer than human experience can easily accommodate, a system in which the present moment is only the latest expression of a continuity extending back beyond familiar measures of time. There is a way to measure absence, not by what is missing, but by the scale of what remains beyond reach. The deep ocean extends across 335 million square kilometers, a continuous expanse larger than all continents combined. Beneath its surface lies a vertical dimension that multiplies that area into volume, a vast column of water totaling approximately 1.335 billion cubic kilometers, layered from the first dimming of light to the seafloor far below. Within that space, most deep-living squid do not inhabit the bottom itself, but the open water suspended above it. They occupy a region that cannot be mapped as terrain, only as depth and movement, where position is defined not by landmarks, but by gradients of pressure, temperature, and faint traces of drifting material. It is within this shifting volume that their lives unfold, largely unobserved. Against this scale, human observation appears in fragments. All remotely operated vehicle dives, across institutions and decades, amount to only a few thousand hours. Each descent illuminates a narrow field, a brief window in a place that extends far beyond the reach of any single expedition. What is seen in those hours represents not a comprehensive view, but isolated moments, each one separated by distances and intervals that cannot easily be bridged. Other forms of evidence accumulate in parallel, indirect but persistent. Hard structures recovered from predators reveal encounters that occurred far from observation. Some of these remains correspond to known species, fitting within established patterns. Others do not, differing in ways that suggest organisms not yet described. The implication is not singular discovery, but the presence of multiple forms that exist without having been directly recorded. Even among animals that have been observed, the frequency of encounter remains low. A squid with arms extending several meters, present across multiple ocean basins, has been recorded only a handful of times. Its rarity in footage does not necessarily indicate rarity in its environment, only the difficulty of intersecting with its path within such an immense volume. Within this space, interactions continue continuously. Feeding events occur. Signals are exchanged. Reproductive processes unfold, each one part of a sequence that has been ongoing for durations far exceeding the period of human observation. Many of these processes have never been witnessed, not because they are infrequent, but because the capacity to observe them is limited. Scientific understanding relies on repeated observation, on the ability to return to a phenomenon and confirm its characteristics. In the deep ocean, such repetition is often not possible. An encounter at a depth of 3,000 meters in a specific location may never occur again under conditions that allow it to be observed in the same way. Each observation stands largely alone, a point of data that cannot easily be replicated. From this emerges a recognition that the current body of knowledge represents only a portion of what exists. Estimates of species diversity remain incomplete, and the identity of many large organisms remains uncertain. The gap between what has been documented and what is present persists, shaped by the scale of the environment and the limits of access. This gap is not an absence of effort, but a reflection of proportion. The domain being studied exceeds the capacity of existing methods to fully explore it. Each new observation contributes to a growing structure of understanding, yet that structure remains surrounded by regions that have not yet been examined. The deep ocean does not withhold itself. It continues as it has, its processes ongoing, independent of observation. The act of looking, limited though it is, reveals only small segments of a system that extends far beyond the current reach of knowledge, a system that remains in motion, whether it is seen or not. There is no final moment in the deep, no clear boundary where one process ends and another begins. Instead, the same movements continue, quiet and uninterrupted, carried forward through water that holds them without witness. What has been described does not conclude. It persists, repeating without repetition, each cycle both familiar and entirely new. Each night, across every ocean, vertical migrations begin again. Small bodies rise through layers of water that grow gradually brighter, following gradients that have guided them for spans longer than memory can hold. Among them, in a coastal inlet that will soon change color with the season, a population gathers in patterns that have returned year after year, independent of any observer who might stand at the shore. Elsewhere, in a region where oxygen is nearly absent, another form drifts with a different rhythm. It does not hurry. It does not chase. It gathers what falls, sustaining itself on fragments that descend from above, maintaining a way of living that requires almost no movement at all. Its presence is steady, aligned with conditions that have remained consistent for ages. In other depths, beyond the reach of most instruments, an unfamiliar silhouette extends downward, limbs arranged in a configuration that does not resemble any common form. It remains suspended, nearly still, interacting with its surroundings in ways that have not yet been fully observed. Its existence is confirmed, its behavior largely unrecorded. Further south, in colder waters, a larger shape moves through darkness with a deliberate motion. Its eyes, proportionally vast, register faint changes in the environment that indicate the approach of another organism descending from above. That descent, driven by the need to feed, connects two forms whose interaction will leave traces only in indirect evidence. Within bodies that will never be recovered, structures accumulate, resistant to digestion, recording encounters that occurred far from observation. Each one corresponds to an organism that existed, moved, and was consumed without ever entering the range of human perception. These records remain enclosed, part of a cycle that continues regardless of whether it is examined. What has been encountered through nets, cameras, and samples represents only a portion of this system. Certain species have been described, their forms and behaviors partially understood. Others have been glimpsed briefly, enough to suggest their presence but not enough to define them fully. Many remain entirely outside current awareness, their existence inferred only through indirect signs. The distinction between what is known and what remains unknown does not separate two different worlds. It marks the limits of observation within a single, continuous environment. The organisms already described do not define that environment. They indicate its edge, the portion that has intersected with the methods available to study it. Beyond that edge, processes continue without interruption. Movements occur, interactions unfold, cycles complete themselves. The absence of observation does not alter their existence. It only defines the boundary of current understanding. What has been gathered so far, fragmentary and incomplete, still carries significance. It represents an extension of awareness into regions that were once entirely inaccessible. Each piece of knowledge, however small, contributes to a broader recognition of the scale and complexity of the system being examined. The deep ocean does not depend on that recognition. It persists on its own terms, maintaining patterns that have endured through vast spans of time. Yet the act of perceiving even a small part of it introduces a connection, a way of acknowledging a domain that exists beyond direct experience. Within that acknowledgement lies a quiet realization. The absence of light does not imply absence of activity. It contains motion, structure, and continuity, extending far beyond what can currently be seen.